All right, welcome back. We hope you're finding the presentations tonight informative. Now I'd like to introduce Dr. Michael Surette, who will present on the human microbiome and fecal microbiota transplant for ulcerative colitis. Dr. Surette is a professor in the Department of Medicine and principal investigator at Farncombe Family Digestive Health Research Institute and has a joint appointment with the Department of Biochemistry and Biomedical Sciences at McMaster University. He is also the Canadian Research Chair in Interdisciplinary Microbiome Research. Dr. Surette is a researcher for the Imaging Network and a member of the team that has carried out randomized controlled trial of fecal microbiota transplant. Sorry, that's a mouthful. Therapy on patients with ulcerative colitis. Please join me in welcoming Dr. Michael Surette.
So it's a pleasure to be here, and thanks to Crohn's and Colitis for the opportunity to talk about some of our research and work that's been done with Dr. Morietti's clinical team. So as a researcher, I'm going to give you an introduction to the microbiome and some of the research that's gone on in our treatment of ulcerative colitis. And consequently, as a researcher, I'm going to educate you a little bit and show you some data. So it's a little bit of a learning experience for all of us. So when we think about bacteria, the first thing that comes to most of our minds are bacteria causing disease and germs. And the germ theory of disease was developed by Louis Pasteur of pasteurization fame. And it was really one of the most important discoveries in modern medicine. And our management and treatment of infectious disease and controlling infectious disease has had a huge impact on human health and longevity. But consequently, it's sort of given a bad name to bacteria. But not all microbes are bad. And I'm going to talk about the human microbiome today, which you may have heard about. This is widespread now in the popular press. This is the cover of The Economist magazine a few years ago, not where you usually go to find your microbiology information. But every other week there's a story in national newspapers and the popular press. So you've probably heard about the microbiome. So what is it? Well, we're all walking ecosystems of microbes, whether we like it or not. And they live on and in us. And this collection of organisms is called the human microbiome. And we really depend on them for health throughout our life and in early life, important for really the development. So this is just a picture to show that basically every surface of the human body, and one thing I should point out, that one way to think about the human body is that the GI tract and the lungs are really exposed to the environment. So they're sort of outside. So we're a tube within a tube. And all of those surfaces in the GI tract and the lungs and the airways, as well as skins and all our mucosal surfaces, are colonized by different bacteria. And what this is meant to illustrate here, each of these body sites has a different community of microbes, and you'll understand this slide a little bit more in a minute, but each of these colors in these pie charts represents a different bacteria, and you can see that what's on the skin isn't the same that's in the throat or in the stomach and so on, because the color is different from site to site. So each body site has really a unique microbiome that plays an important role at that site. So what in fact does your microbiome do for you? So some of the most obvious important things is they produce important vitamins and nutrients that we need that we can't get from other sources. As Dr. Morietti mentioned, when we eat, we first feed our bacteria, and the bacteria provide breakdown products of the food that are actually essential for us that we often can't generate. So they extract energy and nutrients from our food and actually provide us. One of the more important roles they do is they train our immune system early in life and throughout life and help maintain our immune system and keep our immune system in balance. And they're also the first line of defense against pathogens. So if you happen to pick up a pathogenic organism from the environment, the first thing that pathogen encounters when it tries to attack the host are other microbes. And I like to think of it as all of our commensal microbiome, our good microbiome, colonizes all the prime real estate in the body. And microbes have to work very hard to overcome that first line of defense, and that plays a really important role in prevention against infection. So one of the things we do in my lab is we study the microbiome, and the microbiome is made up of hundreds or thousands of different types of bacteria. So how can we study them? Well, conveniently for us, each of these microbes has a barcode. It's a genetic barcode, a sequence of DNA that essentially identifies us as to what it is. And so we take advantage of that barcode to identify and classify the organisms that are present. And so this unique signature really allows us to study the microbiome, and advances in technology in the last few years have allowed us to do this in ways we could never imagine possible even 8 or 10 years ago. So we've really taken advantage of new technologies. So if we look at something like the gut microbiome, we can take a sample, whether it's a stool sample or a biopsy, and we can ask what microbes are present by taking advantage of these barcodes. So we pull out the barcodes and we sequence them thousands or millions at a time with these new sequencing technologies. So what does that really look like? Well, you know, microbes are small, rather oblong or circular things, and I like to think of them as jelly beans. And you can think of the gut as this collection of multicolored bacteria. Unfortunately, they're not multicolored, and we can't tell them apart by looking at them, but we can tell them apart with this barcode. So we use these barcodes to essentially classify these organisms. So basically all we do is we separate them into their groups or colors, and we use this simple separation to identify them. And so we can take this nicely sorted jar of jelly beans and convert it to one of these nice colored bar plots. That makes our job seem really simple and trivial, and we spend a lot of time doing just this. But it's quite informative. And so this is an example of a profile of stool from 2 different individuals. I'm not going to ask you to read or memorize any of the names here. I don't like to say them either. But what I want to point out is that these look different. The microbiome in these 2 healthy individuals don't look the same. And in fact, we all have our own unique microbiome. So if we were to collect samples from everybody in this room and do a microbiome fingerprint, we could come back years later often and do the same thing again and be able to identify each person based on this signature or this composition. So this microbiome is with us. It develops with us in early life, and in healthy individuals persists throughout life, and is really our each own unique microbiome. And this has implications and complications, which I'll mention in a little bit. But what happens when the microbiome goes off when there's problems? If there's disease, and a good example is if we treat with lots of antibiotics, the microbiome is altered. These bacteria are susceptible to antibiotics, just like the bacteria that we're trying to treat, the pathogens. And the microbiome is shifted. And most of the time after therapy, the microbiome bounces back to where it was, to its normal state. So it's robust and resilient most of the time, and goes back to this state. But it is susceptible to these interventions and disease that can really throw it off. Sometimes, however, it doesn't bounce back. It now goes back to a different state, and you can see when it goes to this state on your right, it looks different than the normal state. And we call this dysbiosis. And this refers to an altered microbiome. It's stable and often is, or sometimes is, less diverse in composition, but not always. There's less colors of jelly beans in this case. So how we define a healthy microbiome is actually tricky. It's not about the composition of what specific bacteria are there, but it's what specific bacteria in each individual is in balance with that individual's immune system. And this really is about a balance of those 2 systems. And so it's difficult to just look at a microbiome profile and say a person's healthy or not. And that's one of the challenges we have because every microbiome is unique. When this system goes off and it's in balance, the immune systems become inflamed. That inflammation is driven in part or largely by the microbes that are present. And this inflammation, of course, is behind the disease, the diseases of inflammatory bowel disease. It's the inflammation which causes lots of the damage, but the inflammation is driven by the microbiome. And so most of the use to treat inflammatory bowel disease, treat the inflammation, and not the underlying dysbiosis or imbalance of the microbes in the immune system. So the question is, can we fix a healthy, can we use a healthy microbiome to fix this dysbiosis? Can we use a healthy microbiome to fix this imbalance? And this is this idea of using fecal transplant to treat ulcerative colitis, or as you heard, potentially Crohn's disease. And this is as simple and as straightforward as it sounds. This is taking fecal material from a healthy donor and using it to treat a patient. We're going to talk a little bit more about that. This is a therapy that's been around for a long time, not practiced widely, but around for a long time. But why it's gained a lot of attention more recently and has really become something we're looking at much more enthusiastically has to do with the treatment of something else, and that's treatment of C. difficile infections. And C. difficile is an example of a bacterial infection that happens when you treat with antibiotics. So remember, when you treat with antibiotics and that jelly bean jar becomes half full, in the case of C. diff, that fills up now with C. difficile. And you get C. difficile by treating with antibiotics, and too many antibiotics disrupts that microbiome and allows an opportunity for C. difficile to take over. And our primary treatment for treating C. difficile is more antibiotics. And you can see that there's a little bit of a disconnect there. And what's been found, though, is using FMT, or fecal microbial transplant, to treat C. difficile, particularly recurrent C. difficile infection, has about a 90% success rate, compared to about a 30% success rate with our best other drugs. And so this is now the recommended treatment if you have recurrent C. difficile infections. And so the idea is you're using microbes here, healthy microbes, to restore or correct the defect in C. difficile, and the rationale is, can we use it to correct other dysbiosis? So this has been around in Crohn's disease and ulcerative colitis for a little while. The first case report was in 1989. There's been a number of small case studies, and a survey of those studies by Dr. Moietti's group showed that there is some evidence that this might work. Now, as you heard, there's many more studies going on, but the first randomized controlled trial was done here and led by Dr. Moietti's group. And we were fortunate, with the support of Crohn's and Colitis Canada, to be able to try and study the microbiome in those cases to try and understand what's going on. So the first successful randomized controlled trial, as I mentioned, was led by Dr. Moietti. The patients had moderate to severe disease. They were treated once a week for 6 weeks with FMT. This was a randomized controlled trial, so there was a placebo group that just received water. And I'm going to come back to that in a minute and try and convince you why that's really important. And the primary outcome of this study was remission at the end of the trial at 6 weeks, which I'll say is a pretty high bar for a test like this, for a trial like this. And so, in fact, when the results of this study was published, there was a second study from the Netherlands published at the same time. In our case, there were 38 patients in the treatment group, and they had 23 patients. The response rates in both of those was about the same, 25% in both cases. So this looked pretty comparable between the two groups. But what I want to point out is there's a placebo group in both of these trials. And in our group, the placebo response rate was very low. And in the other trial, the placebo response rate was the same as the treatment group at about 25%. So one of the questions that's often asked is, why don't we just treat everybody and see if the process works? And this really illustrates it. This high placebo response rate made that study non-significant. So why was there a high placebo response rate? This study had milder patients in the treatment, and just the natural cycle of flares coming and going means a lot of patients had improved symptoms over the course of the study. And this gives this higher placebo rate in this patient group really gives gives the impression in the treatment group that it's working when in fact it doesn't. So I really want to highlight that this is really important, and this is why we need to do these randomized control trials to understand the two groups. So again, the remission was about 25%. Using these same criteria, this is comparable to the remission that you get with our biologics. So this is comparable to the best alternative therapies that we have at the time. There's an interesting sub-story in this study in that 7 of the 9 people responded with the same donor. And this donor in some circles has become quite famous as the super donor. This we're still trying to understand and figure out. But this was a successful study. But as you probably gathered from Dr. Moyetti's, 25% response rate is not what we want. We want much higher than that, and we need to go on. And I'm going to come back to that in a second. But I'm going to now go back and talk to you about a case report. And this is, in fact, a study that wasn't represented in the responders. And I'm going to go back to some of the things that we learned a few minutes ago and walk through some of the data. So this is a patient that had ulcerative colitis for 16 years, severe disease, went into the trial, and was randomized to placebo. These are randomized and we have no control, and unfortunately this patient ended up in the placebo arm. At the end of the placebo arm, there was no response to treatment, and the patients are allowed to enter into treatment independent of the study. And after 6 weeks, he showed some significant clinical improvement, but didn't go into remission. So had this been in the trial, this would have been, by our criteria, still a failure, even though his symptoms improved. And after 15 weeks of treatment, he went into complete remission and stayed into remission for about 5 months with no further treatments of any kind. So now remember back to the jelly beans and the colored bar charts. This is what the microbiome looked early on. So on the left here, I'm going to have to point to this. So this is at the very beginning, and this was the first 6 weeks of treatment. There's really not a significant change in the microbiome. And then as he goes on more and more treatment and ultimately gets better, the microbiome changes. What you might notice is the red bacteria decreasing, the orange bacteria are increasing, and the blue bacteria going down. In general, this group of bacteria are things that we think of as good bacteria, so they seem to be increasing. This is a group that we don't expect in the GI tract, and we like to see that go down, and the same with the other group. So there is a change in the microbiome that goes on. It's shifted to something that we would think of as a better microbiome and improvement, and we think that's part of the therapy. I will point out, though, that this orange bacteria that increases was present before he received any treatment, and the question of whether or not he's actually getting that bacteria from the donor or just his own good bacteria is being restored is an open question that we're really trying hard to understand. Unfortunately, after this 5-month period of being in remission with no treatment, the patient relapsed. We don't know what the trigger was, but again, if you look at that composition of the microbiome, the orange bacteria went down, the red bacteria, the blue bacteria came back. But again, with further FMT, it was restored again, and the patient went back into remission. So what did we learn from this study outside of the placebo study? First is that the placebo really had no effect on his microbiome, as we expected. That the FMT really did work. It took a longer course of time than in the clinical trial, but the microbiome did change and the patient did go into remission. Remission was sustained for a while, but a relapse did occur. At least in severe UC, this probably means that these fecal transplant studies aren't a cure, but a treatment that can induce remission. Maybe over a longer term, we wouldn't see these potentials for relapse, but this is something we need to understand more of. And these good bugs that were restored, many of those seem to be present before the treatment, and maybe what we're doing with this treatment is somehow making the patient's own good bacteria come back as opposed to get them from the donor. And this is actually a much harder problem to sort out than it sounds, and we're working very hard to do that. So how can we improve this? And this is the goal of the next study, and Dr. Moyetti mentioned this in his talk. So the new trial's underway. In this case, all the patients are being treated, but now they're randomized to a pretreatment or not. And that pretreatment is to pretreat with antibiotics and see if that will improve the efficacy of the treatment. Again, we're using a single donor, that donor B. The therapy is longer duration. It's 2 times a week for 8 weeks. So this is a highly inconvenient treatment, but the frequency seems to be really correlating with efficacy. And in other studies, those that treat more and more often seem to have more effect than those that don't treat very much. And again, this is being led by Dr. Moyetti's team here at McMaster. Some final thoughts about UC. And as you heard earlier, FMT is not without risks. Donor stool has to be very carefully screened against pathogens. We all carry around pathogens all of the time in our healthy microbiome. And the rest of our microbiome and immune systems keep those in check. So just because someone's healthy doesn't mean they're a good donor for FMT. And this is one of the reasons that we really extensively characterize these patients before using them for FMT. So it's a treatment not without risk. I would say that we still don't understand how it works. If we understood how it worked, I think we could make it better, and so that's one of the things we're actively trying to sort out. It does work better for some people than for others, and is that something intrinsic about the patient, or is it something intrinsic about the donor samples that we're using? How do we understand that? And again, larger randomized control trials help us get at these questions. And then a couple final thoughts about the microbiome. Remember that the microbiome and the host, you want to think about these 2 things. The immune system and the microbiome is being really integrated. We have individual microbiomes. We have individual immune systems in many ways that are tailored and shaped by our own microbiome. And these interactions between the microbiome and the immune system really play a major role in controlling health. As you heard earlier, they do affect our mental status and mental health, and that's an important aspect of the Imagine network, trying to understand that aspect of it as well. The other thing to remember is that all of the things that we do, as we mentioned earlier, our diet, when we eat, it's impacted by the microbiome, it's digested by the microbiome, and it's presented to the immune system and the rest of us through our microbiomes. So that impacts us in very individual ways. Probiotics are other microbes that impact this system through the same way. Drugs that we take, many drugs can actually be metabolized by the microbes that we have. And there are now many examples where somebody may not respond to a particular drug where many other people do, and it turns out they have bacteria that are degrading that drug. And so that's another kind of interaction that we need to understand. And again, how behavioral therapy and treating some of these other comorbidities not only can treat the psychiatric comorbidities, but may, in fact, through the microbiome affecting the immune system affecting the microbiome, alter the state of disease. And these are important considerations that we need to look at. And one of the things that the Imagine network and using the ability to look at large numbers of patients is really going to allow us to do. And so finally, I'd just like to highlight that our research has been made possible by Crohn's and Colitis, and this study has been made possible by the wonderful collaborations with Paul Moyetti and his clinical team. And we're looking forward to pushing this data harder and understanding this more in the second trial. And understand this process, understand how this works, and use that understanding to make the efficacy of this treatment better, and hopefully make it a successful therapy for more patients and make it safer. So thank you.